Hey guys, welcome to the review for Exam 3 for Bio 246. This exam, it seems like there's going to be quite a few chapters with a lot of information within each chapter. The chapters that are in the exam are going to be viruses, antibiotics, pathogenesis, and immunity. I do not know exactly where you guys stopped when it comes to immunity, so I'm going to go over pretty much what the study guide says, and if you did not go over in class, ignore it. It will be on exam 4. So, like I said in the previous two videos, the review is a great study tool, but do not just rely on that. Please study your PowerPoints. The PowerPoints are your best help. Please read the book, as the book will give you pretty good information, but the PowerPoints are your best source of information. So just go through those, just look over everything. Like I said in the other two videos too, it's a lot of information to read, it's a lot of information to go over and memorize, but just think, there's only about four more weeks left, about 30 days. That's including Thanksgiving. That's also including any days that you have in between your finals for finals week. So we're almost there. So if we just want to dive right into it, viruses. Yes, viruses is a very difficult chapter. Viruses is a lot of information to remember. My best tip for you guys to study this, I know I put this on Piazza, for you guys that are on Piazza, just memorize is the best. Go over it. So hopefully you guys have started studying about a week ago. That would be the best bet to this chapter. I really have no other information or study tips for this, because what I did when I was in Bio 305 is I just studied this material over and over and over again. I did make flashcards. I know flashcards aren't for everybody. I made them, and at the time I lived in a dorm, so I just kind of taped them to the wall, and I would go by and I would look at them every day. I know some of you guys either live at home or you're unable to tape stuff on the wall, and flashcards sometimes don't help people. I get it. So the best thing is just memorization. It's the best tool you have in this class. I've got a couple questions on Piazza. What do we have to know exactly on the slides for viruses? And my response, I know is not a response that they wanted to hear, but it's everything. As you guys saw on the exam 2 for bacteria, Dr. Kokash was and did ask any question in the book about bacteria. You will never know what virus she's going to ask, and you don't know exactly what specific characteristic of a virus she can ask. So you have to have everything in the back of your mind, so when you do see the question, you can pull it right out. Definitely, if anything, know if it's double-stranded or single-stranded. Know if it's DNA, RNA. Those are the big ones. So just know if they're enveloped or non-enveloped. That's another big one to go over. So those are the three characteristics that I would know the most, as well as, of course, the name of the virus. If y'all want to pull up the PowerPoint for viruses, and as I know, as you already did the video, you probably noticed the Ninja Turtles and the weird curtains in the back. I bought my house in March, and this is one of the bedrooms. It's the spare bedroom. It's where I throw everything in. Like I told you in my email, I just got hardwood floors downstairs, so I kept the video at an angle where you can't see the floor, because there's stuff all over this floor, because I have yet to rearrange the hardwood floor, because it took 48 hours for the stain and the shine to finish. So it's, I've been traveling outside to get to my kitchen and back and forth. Anyways, so that explains the Ninja Turtles, and the curtains were the old owners, and I've just never taken them down. So anyways, back to the virus. So the first couple of slides are pretty much background information. It's the history. I would know it because it could pop up. I know that Dr. Kokash has put some of the main important people on the exams before, so I would know it. I would know what virus is in Latin, which of course in Latin it's the first sentence is poison. Know that the size of the general characteristics, which is the size, they're very small. They're actually filterable, so they can actually go, if it's 0.3 micrometers, it can actually fit through the holes. It needs an electron microscope to see. It may or may not have lipid envelopes. That's one of the characteristics you need to know. Is it enveloped? Is it non-enveloped? And of course, it has a range of host cells that it can affect. Know that it can contain DNA or RNA. That's another one. And also it's a single-stranded or double-stranded. I would know that they, like I said, on slide 6, there's classifications. The nuclear content, DNA, RNA, the envelope, stuff like that. I would know the categories of the viruses. I would know them, which you can find those on, I believe this is slide 7. You can find the herpes virus, the retrovirus, the adenovirus. It gives a good little chart of the enveloped and the non-enveloped, so you have a good idea of it going into the PowerPoint with the rest of the list of viruses. Then you'll get single-stranded, double-stranded. Like I said, it's pretty much memorization from here. Know if it's double-stranded, single-stranded, DNA, RNA, and is enveloped and unenveloped. I know I keep stressing that, and I keep repeating myself, but once again, that is a big takeaway in this chapter. So yeah, just going through the chapters. There's a lot of viruses that are going to pop up. If Dr. Kokash mentioned them, I'd know them. So I'll give you an example. The adenovirus. It is on slide 27. Sorry guys, my nose is really itchy tonight. It's number 2. It gives a category of dsDNA, which that means double-stranded DNA. It's ice. That gives the shape next, and then it says naked, which means there is no envelope. And then it just gives a short definition of what that virus is. It's a respiratory disease virus. If I go through another example, I'll give you poxvirus. Once again, dsDNA, complex, and enveloped. So that means it has an envelope. So it says naked, non-enveloped, enveloped, it has an envelope. And of course, poxvirus is an example of a virus that has a category of chickenpox or smallpox or cowpox. Let's see, I'll give you another example. Here we go. Togaviridae. It's positive RNA and it's enveloped. And good examples of that would be rubella, which we all know from the controversial MMR vaccine, measles, mumps, and rubella. Do know that it could pop up on this exam. If it says German measles, that is not measles. German measles is rubella. If it just says measles, that is the measles that we all know. But once again, if it says German measles, that is not measles. That is rubella. So yeah, going through, it's just more of the same. Unfortunately, I know it's memorization, but there's only 4 more weeks left, guys, and this is really going to help further on in your career. These chapters are chapters that you're going to use for the rest of your career. As a nurse, as a member of a global health organization, you're going to need to know this information going further. I don't think there's much else I want to mention. Let me just go through the PowerPoint. Oh, yes. Yes, there actually is. The viral replicative cycle for viruses. Please know the list, and please be able to list this. Dr. Kokash can give this question in a multiple, a bunch of different ways. I've seen it written a bunch of different ways. She can give you a list, 1 through 6, and say 1, 4, and 6 are filled out, and you have to fill out 2, 3, and 5. It's also where she could give you a word problem, and it might say the list, and it might skip 1. Or it might give you 1 through 5 and say, what is 6? So know this list. I'll give it to you right here. 1 is attachment. Of course, you won't be able to replicate anything if it does not attach. So number 1 is attach. Then that attached has to penetrate. Then it uncoats. Then it's biosynthesis. Then it's maturation. And then it's release. Once again, that list is attachment, penetration, uncoating, biosynthesis, maturation, and then release. These are the steps to the viral replicative cycle for animal viruses. And then it gives you in the PowerPoint a good breakdown of what each of the 1 through 6 is. Know that as well. There's also one for retroviral replication, which retroviral we all know as a retrovirus, which the most common one that we all know is most likely AIDS. Know this replicative cycle. It is similar to the viruses, and the retroviral replication goes as this: attachment, penetration by fusion, biosynthesis, assembly, release. Then it goes from RNA to DNA to RNA. So let's compare those two. So the replicative cycle for animals was different than the retroviral replication for retroviruses. The one for regular animal viruses goes as attachment, penetration, uncoating, biosynthesis, maturation, release. And then if you want to go to the retroviral, it's going to go similar, but there is a step that is different. The step for this retroviral goes attachment, penetration, biosynthesis, assembly, release, and then RNA, DNA, RNA. So when comparing these, the retroviral replication is missing the uncoating step that was involved in the animal virus replication. And then if you compare the two again, even though we see the retroviral is missing the uncoating step, we see that it added a step going from RNA to DNA to RNA, which the regular replication for animal viruses do not do. We also know viral latency. They can be latent, which means that the virus is inside of your body, but it remains quiet, quote unquote quiet, until something activates it. The best example for that is if you have had chickenpox. You have the chickenpox virus inside of you, and it could resurface as shingles. So shingles is the virus that remains latent. It's HSV1, or simple X virus. HSV1 is what's most commonly known as. I would know that viruses and cancer can relate in a simple way. There are some viruses that can cause cancer. The 3 ones are EBV, which is Epstein-Barr virus, HPV, which is the human papilloma virus, which we all know can be associated with cervical cancer, and then there's HBV, which is hepatitis B, which is associated mainly with carcinoma, which is liver cancer. So I would know those 3: HBV, and HPV. I know it's a little confusing, but it's good to know those. Also know that retroviruses can cause leukemia, which we all know if within HIV and AIDS it attacks the white blood cells, which leukemia is a cancer of the white blood cells. And then if you go to the, slide 65 in the virus PowerPoint, it gives you examples of prions. I would know a few examples of prions: Creutzfeldt-Jacobs disease, mad cow disease, and scabies. I would know that where scabies can be found, which is found in sheeps. Know that prions are infected particles. They're the protein only. It can go to the brain, and it converts normal protein to an infectious abnormal protein. So once again, prions. Just know a few examples: Creutzfeldt-Jakob, mad cow disease, and scabies, which can be found in sheep. And it could go to the brain, and it's the infectious particle that turns normal proteins into abnormal proteins. I think that's all I want to say for viruses. Antibiotics. This is another chapter where you just have to study. You have to study. You have to know between each subcategory which antibiotic is subcategorized into that. So if you look at the review guide that Dr. Kokash has put up, she was nice enough to give you a wonderful list that I would know inside and out. It is not the entire list, and I would not just focus on this. I would focus on the PowerPoint as well. The PowerPoint gives you more information, but Dr. Kokash laid this out for you, which I think is very, very nice of her. So as you can see, there's a few questions at the top. What is an antibiotic? Selective toxicity. Understand how resistance works. We all know from the concept map that was recently turned in that you had to list 4 reasons of how resistance occurs. What you could say a multiple of things: rapid efflux, not using the antibiotic right, which means not doing the full length of the antibiotic drug, overuse of the drug, taking it too much. There's a multiple things you could say. If you did not get that on the concept map, please study this. I can promise you it will be on the exam. So there is 6 questions before the list, so definitely know those. But the list is what I think is really important. She broke it down into the subcategories, which the subcategories are inhibitors of cell wall synthesis, 2, inhibitors of protein synthesis, 3, injury to plasma membrane, 4, most antiviral drugs inhibit the nucleic acid synthesis, and 5, inhibitor of metabolites. If I just go through the list quickly, inhibitors of cell wall, of course we all know penicillin. Please know that there's different kinds of penicillin. Please know that there is natural and semi-natural penicillin. Know that the difference between the penicillins are the R group. Please know that there's a multitude of different penicillins. I'm sure we're all familiar with of course the natural penicillins, and then we're also known to the semi-natural, which are made in the lab, which we are all pretty much familiar with amoxicillin if you have ever had an infection before. Of course, we all know who just stumbled upon penicillin. So just know that cephalosporins. Sorry, really late at night. So those are the 2 main inhibitors of cell wall synthesis. Inhibitors of protein synthesis: no tetracycline, macrolides. There's 2 others. The main one I want to point out is tetracycline. Please go back to your PowerPoint and notice that there is a specific thing with tetracycline. It does make teeth brown. So I've seen that on exams before. I do not know it's on this exam because I do not see the exams beforehand. I see the exams when you guys see the exams, but I have seen it on the exams before. Subcategory 3 is injury to the plasma membrane. There's Pauline, there's azole antibiotics. I would know those. Know that they inhibit plasma membranes of fungi. So there's a lot of fungus involved. So know that. Of course, antiviral drugs. We all know AZT, which is the most famous drug for individuals who unfortunately have HIV or AIDS. It is one of the most common drugs for that virus. know that they are very toxic, and there are very few other drugs besides AZT that work to inhibit nucleic acid. And then we have inhibits the metabolites. So you'll see that there's 2 of those. Pretty much they inhibit the folic acid synthesis and its analogs for PABA. So like I said, know the list. It's not that big of a list. You have a lot more viruses, unfortunately, to study than the antibiotics, but know the list. Just know what subcategories they go in. Know how each antibiotic affects that subcategory it's in. So know how penicillin affects or inhibits cell wall synthesis. For the next section, I see it's pathogenesis and immunity. I see that Dr. Kokash has combined those PowerPoints. Like I said, I do not know how far you guys have gotten into this, so I'm just going to go ahead and review what I think is important in those 3 PowerPoints. And like I said, if you did not get to it in class, ignore it. I will figure it out, and I will reiterate the information that will be on exam 4, or the final exam review session that I'll make. So if we just want to go right into it, I'll just hop right into pathogenesis. That's PowerPoint. The first couple of slides are good definitions. Just know, disease, infection, etiology, pathology, what they are. Of course, pathology is the study of a disease. Etiology is the cause of the disease. Note the host is where the organism resides. Normal flora, know that there's resident flora and transient flora. Transient flora is the flora on your hands. When you wash your hands, it comes and it goes. Resident flora is always present on the human body, and it does not get removed. Know about, of course, the Human Microbiome Project, started in 2007. Is a project that was used to identify all of microbes and the relationship. That was a huge project, so definitely know that, which can be found on slide 4 of the PowerPoint. Symbiosis, know that is the association between the 2 or more species, usually it's 2. No, commensalism is one benefits, the other one is not affected. So an example of that would be Staphylococcus epidermidis, which is on your skin. It's a normal flora, it resides there. So the host benefits and it presents pathogens from overgrowing. So pretty much one grows and thrives, and the other one is not affected at all. Mutualism, both benefits, which is the coli in the intestines produce vitamin K and some B vitamins, which we know we cannot live without vitamins K and B. And then we got antagonism, which is competition between the microorganisms, which is bad. It does not benefit anybody. Parasitism is one organism benefits, the other is harmed. Let's see what else. Opportunists. An opportunistic organism do not usually cause a disease, but can become pathogenic under certain circumstances or environmental conditions. No, I see this on pretty much every exam. The exam I took in Bio 305 and the previous exams that I have been the LA for Dr. Kokash. Know what symptoms, signs, and syndrome are. Symptoms are subjective, as changes in the body such as pain. Signs are objective. They can be seen by everybody else. A rash, a fever, swelling. Those are things that can be seen by other people, so they're objective. Remember, symptoms is subjective. It's pain. No one else can see it but you. You're the one who feels it. I can't feel your pain for you, but I can dang sure see your sign, which I can see you have a fever. I can see you have a rash. And then syndromes are specific symptoms and signs that are associated with the disease. So say you have the flu, you have a fever. Of course, that's a you see that within the flu is a fever. So it's specific symptoms and signs associated with the disease. Know the types of diseases. They can be infectious, which is caused by bacteria, viruses, fungi, protozoa. They can be non-infectious, caused by any other factor inferior to organisms. Incidence, prevalence. No, prevalence is the total number of people affected within a population. I see this on exams as well. Sporadic, endemic, epidemic, pandemic. Sporadic occurs occasionally in a population. It happens every once in a blue moon. Endemic, a disease that is always found in the region. Epidemic, a disease that many people acquire in a very short period of time. And pandemic, which is a worldwide epidemic around the world. So, once again, sporadic happens every once in a while. Endemic, a disease that is always found within that population. Epidemic, which is a disease that happens to a bunch of people in a very short amount of time. And then pandemic, which is that epidemic but worldwide. This is also on exams: acute, chronic, latent. Earlier in this video I mentioned latent as the shingles virus. Latent means just, of course, the virus was introduced to your body and it remains quiet until it is activated again. Chickenpox, the virus remains in your body, it gets activated, shingles. Chronic is a long duration. You can have it forever. A good example of that is AIDS. It's in for the long haul. Also, leprosy, which is an example that Dr. Kokash gave in the PowerPoint. Acute is just a short duration. It comes and happens. It shows its presence and it's gone. A good example of that is the flu. The flu hits you hard, but just for a short amount of time. The spread of the disease. The reservoir is the source of the disease causing organisms. It can either be living or inanimate. It does not have to be living to pass along or be a reservoir for a disease. Of course, living reservoirs, you have human reservoirs, which may transmit the microorganism directly or indirectly. Then you have animals, which are wild or domestic. Zoonoses is a disease that occurs primarily in animals, but can be transmitted to humans. A good example is bubonic plague. It was in rats, but of course it was transferred to humans. Lyme disease is another one. It's a zoonoses because it happens in ticks, but of course ticks can transfer Lyme disease to humans. Then we have non-living, which is soil. That's where you get a lot of your fungal diseases from. Water, which can be contaminated by feces, which is typhoid fever, E. coli. Those are just a few examples of diseases that you can get from the reservoir, non-living reservoir of water. Then you have your modes of disease transformation. You have direct, which is person to person. I personally give it to you. That can happen through sexual contact, kissing, touching one's sores, shaking hands, or the fecal-oral route. It can also happen from parent to offspring. It can happen between the egg and the sperm, breast milk, placenta. So it's pretty much just direct human-to-human contact. Indirect are fomites, which are syringes, drinking glasses, anything that is a non-living object. And then you have droplet transmission, which is droplet as in sneezing. I sneeze, drops or droplets get on you, and that is a mode of disease being passed along. No, it is not direct because it's not person to person. I'm not kissing you. I'm not being intimate with you. We are not holding hands. We're not shaking hands. I literally just sneezed on you on accident, and droplets of my mucus or my snot have gotten on you. For droplet transmission is a very short distance. It does not go long distances. It has to be short, as in pretty much up close and face to face. Dr. Kokash gives you pretty good examples, I see, of pictures. As you can see on slide 28 of the mechanisms of pathogenesis, that's a good example of droplet transmission. The young lady is sneezing, and of course the droplets are coming out. The vehicle transmission can be through, this was another problem on your concept map. the vehicle transmission is going to be water, food, and air. Water is usually cholera. It's due to fecal matter being added to your water. So say sewage somehow gets into the drinking water system, that causes cholera. Food, botulism, we all know, do not eat a can that is swollen, and that's been in your pantry for a while. And then air, of course, once again, mucus droplets. I sneeze, it's now in the air. A good example of that would be tuberculosis. So those are the vehicle transmissions. Please know those. Dr. Kokash also gives you pretty good examples of pictures for those on the slides. Then you can be biological or mechanical. Biological is if a person is bitten by insects that are harboring that microorganism. The microorganism replicates inside the vector. A good example of that would be malaria or Lyme disease. An animal biologically bites the human, and of course the virus is then replicated, or the microorganism is then replicated inside of the vector. Then you have mechanical. Is transferred the microorganism from the feet to other body parts. So say you're walking and you don't have your shoes on and you touch the bottom of your feet randomly and you forget to wash your hands and you begin to eat. It's passive, and then of course there is no replication of that vector, but you did just transfer the microorganism from your feet to the other part of your body, from your hands, of course, to your mouth. Once again, there is pretty good examples of that Dr. Kokash gives you. The next slide is nosocomial infections. Nosocomial infections are the result of staying in the hospital. We all know a big issue is MRSA. You see that in the hospitals. Dr. Kokash gives you a statistic, which is 5 to 15 patients are affected by nosocomial infections, which means it's about 2 million or 10% in American hospitals, and has increased within 36% in the past 20 years, which shows that it's crisis. It's bad. So there's 3 factors: the microorganisms in the hospital, there's a compromised patient, or there's a chain of transmission within that hospital. When it comes to the compromised patients, usually the host is immunocompromised. There's a resistance to the infection is reduced, so that means broken skin. You have surgery, you have burns. You just came out of the OR and you're in the recovery room. Of course, you have open skin. You have skin that was just recently stitched or stapled shut. And of course, there's going to be microorganisms that are going to take advantage of that because your immune system is now immunocompromised. So it is opportunistic, and that's how that it can pass through compromised patients. When it comes to the chain of transmission, there's a bunch of them. Fomites, which remember are the syringes to the patient to patient, hospital staff to patient. You have a million different hospital staffs between the doctors, the nurses, the med techs, the people that are coming to change the dressings, the housing staff that are coming to get used or soiled clothing, cleaning the bathrooms. There's a bunch of people going through, so it could always have the chance of spreading microorganisms, so it becomes a chain of transmission. There's a few ways you can control nosocomial infections. Washing hands is a simple one. Just wash your hands. You see doctors now, when they leave every patient room, there are the hand sanitizers they use. They put it on their hands to hopefully stop the chain of transmission of nosocomial infections. There's a bunch of emerging infectious diseases. Of course, there's many different factors. The diseases that are new or changing every day. We have a lot of antibiotics that are now becoming resistant to the disease, so the disease no longer accepts the antibiotic because it's now resistant, which again, we talked about that, how it became resistant. You didn't use your antibiotics to the entire course. There's many different things. there's an establishment of a disease. So I gave you a list of the way that the viruses and the retroviruses replicate. There's also a list of the establishment of the disease, which there has to be a portal of entry. It has to adhere to the tissues. If there, once again, like I said, if there was no attachment in the replication and retrovirus cycles, there would be no cycle. There would be no replication of that virus. Same with the establishment diseases. If it does not enter and it does not adhere, it's not going to happen. It's got to penetrate the tissues, and that is going to cause damage to the tissues. There's different portals of entry. Mucous membrane, the skin if you have a cut or a wound, the parenteral route, which is bites, punctures, burns, injections, and so on and so forth. Of course, adherence. We all should know what biofilms are, which is just the masses of microbes. It's a community that lives together, microorganisms. There's, of course, a different bunch of portals of exits. There's a urogenital tract, which is sexual contact with urine. The gastrointestinal route, which is vomiting or fecal removal. Blood, and respiratory tract. Let's see. Of course, bacterial damage to host. Toxins are poisonous substances produced by that microorganism. There's exotoxins, which is produced inside bacteria and released to the surrounding medium. And endotoxins are lipid A portions of LPS that is released from the cell wall of gram-negative bacteria. Because we remember that gram-negative bacteria is the ones that contain the lipid A. And then if you go through the rest, there is bacterial endotoxin, which goes over endotoxin itself and the two possible outcomes, which are fever and septic shock. And the rest is pretty much the exotoxins and endotoxins. Study that, know what they are. There's actually a really good slide on slide 76 that compares and contrasts exotoxins and endotoxins. I would know that chart for your best benefit. And then really quick, I'm going to go over the immunity chapters. First, I'm going to go over non-specific. So of course, there's a first couple PowerPoint or slides in the PowerPoint. There we go. As terminology, which is resistance is the ability to prevent a disease occurring, which is of course known as immunity. Susceptibility is the amount of vulnerability to a disease, so which means the lack of resistance. Sorry, there's innate resistance, which is not specific, and adaptive, which is specific. We'll get into adaptive the next PowerPoint that I talk about. Slide 3 is a really good slide. It breaks down the immune system between adaptive and non-specific. So non-specific contains the first and second line of defense for your immune system, and then of course specific or adaptive is your third line of defense. For non-specific, the first line of defense is the intact skin and your mucous membrane. The second line of defense is phagocytosis, inflammation, fever, and antimicrobial substances. So breaking it down, the innate immunity is present at birth. It does not have a memory. So when these lines of defense see a virus and it defeats that virus, there is no memory of that virus, and when it comes back again, it will have to fight it again because there is no memory. So remember, non-specific has no memory, and later on we'll talk about how adaptive specific does have a memory. So like I said, the first line of defense is intact skin. No cuts, no wounds, no burns, no surgeries, and your mucous membrane, which lines, of course, your GI tract. There's mechanical barriers, which is your mucus, your cilia, which is your nose hairs, Their cilia can also be found in a few other places, your tears, your saliva, and the flow of the urine that cleans the urethra and vaginal entrance. There's chemical factors involved as well. Gastric juice, which of course we all know has a very acidic pH. Vaginal secretions, which also has a acidic pH. Perspiration, which eliminates and washes and flushes away the microorganism. Stomach acid, which of course most bacteria kill due to very low acidic pH. And transferrin, which is an iron-binding protein in the blood that prevents bacteria from utilizing the all-important iron. Your second line of defense is inflammation, fever, and antimicrobial substances, which is interferon and the complement system. Of course, you have different kinds of cells. First, we're going to talk about granulocytes, which are neutrophils, which are 60 or 70% of the actual circulating white blood cells, and they are highly phagocytic, or involved highly in phagocytosis. Basophils, which is only 0.5 to 1% of your circulating white blood cells, so not a lot. This one is involved in inflammation and allergies. Eosinophils, 2 to 4 of your circulating white blood cells, and these are also involved in your allergies and may be phagocytic. Then you have your monocytes, which are 3 to 8% of your circulating white blood cells, which is your macrophages, which are wandering. Monocytes processes antigens, which is a substance that can elicit antibody response. And of course, dendritic cells are derived from those monocytes. Then you have lymphocytes, which is 20 to 25% of your circulating white blood cells. These are natural killer cells, and you have 2 B lymphocytes, which produce antibodies, and T lymphocytes, which is involved in cell-mediated immunity. So if you go back through these that I just mentioned, the highest ranked one is neutrophils, and the lowest ranked one is your basophils. So I would know that. I would be able to put all of those in order based off the percentage of circulating white blood cells within them. And then it goes into a slide about phagocytosis, which means to eat, which is the ingestion of a particular matter. Then there's leukocyte, which of course are the white blood cells. There's 5 basic types: 3 granulocytes, monocytes, and lymphocytes. Once again, I would know the percentage of those, and I would be able to rank them in order, either from highest to lowest, lowest to highest. And then of course we know is leukopenia, which is decrease in white blood cells, which I mentioned earlier in this video is AIDS, and then leukocytosis, which is the increase of white blood cells, which is like gonorrhea. Then there's the mechanisms of phagocytosis, which are chemotaxis, which is the movement of the phagocyte to the site of the infection. And then we have adherence, which is the attachment to the phagocyte to the bacterium itself. Ingestion, digestion, and then there was a residual body, which is the non-perishable or non-digestible material that is discharged from that phagocyte. Then we get into inflammation. Please know these slides. This is not only important for this class, but important for your future career. Know the process, know about the vasodilation of the blood vessels, the phagocyte migration, the phagocytosis, the tissue repair. I would know it all. If you need to study those slides, they start on slide 25 of the non-specific PowerPoint. Please go over those, definitely. Dr. Kokesh gives you really good pictures and step-by-step of what happens in the process of inflammation. Please go over those. Of course, fever is a systemic response to the infection. There's shivering and chills involved, and it's actually beneficial to the host if it's controlled. I know that sounds crazy, but fevers can definitely be beneficial to the host, but only if it is controlled. Then we have interferons, which is the class of small proteins produced by some animal cells after a viral infection. Of course, interferons interfere with the replication of those viruses. There's 3 types: alpha, beta, gamma. Alpha is produced by B lymphocytes and monocytes and its antiviral activity. B is produced by fibroblasts, and gamma is produced by T cells. So I would know which is produced by what, where alpha, beta, and gamma, where it is produced at. Know the complement system. This starts on slide 38 of nonspecific. Know this. This is very important. Also, know the pathways that are involved, which are classical, alternative, and the lectin pathway. Those are all part of the complement system. Know those 3. Once again, classical starts on slide 41 of the nonspecific, and it goes all the way down. Then it goes alternative, then it goes to lectin, and it stops on slide 55, which is the end of the PowerPoint. Pretty much this PowerPoint is shorter than the others, but there's a lot to know when it comes to the process of inflammation. It comes to the complement system. It comes to classical, alternative, and lectin pathways. When it comes to white blood cells, the 3 granulocytes, the monocytes, and the leukocytes, knowing the percentage of each. So just please, hopefully you studied that. If not, you have Wednesday, you have Thursday up until your exam at noon, and you have Dr. Kokash's review session at 3 p.m. tomorrow afternoon. If you cannot go, please go. But these are definitely a time to take your questions from this video and ask. Get clarification if there's anything. Of course, my Piazza's up and running. So is my email. And I want to mention that as I go into the very last chapter, which is specific. Specific, as I said, is your 3rd line of defense. There's a bunch of information in this one too. It's the shortest PowerPoint of them all, but there's a lot of information. Right off the bat, you need to know slide 4. I've seen this on every exam. Naturally acquired active, naturally acquired passive, artificially acquired active, artificially acquired passive. Know an example of each. So naturally artificial active. Person acquires the immune naturally when it carries a pathogen. Naturally acquired passive is transferred between mother to fetus. Artificially acquired active is receiving a vaccination, and artificially acquired passive is immunity acquired after injection of performed antibody. The humoral immune system is the B lymphocytes, cell-mediated T lymphocytes. Of course, now we're going to get into the antigens, which are extremely important. Please know them all. Know the antibodies. Then we're going to get into the 5 classes of the antibodies. Between the heavy chain and the light chain. There's a good chart right here on slide 14. Know it. Know it, know it, know it. You're also going to have to know the percentages of these bad boys right here. IgG, IgA, IgD, IgM, IgE. You're going to have to know the 5 of them. Know that IgG is the highest and IgE is the lowest. To break it down, IgG, it's the monomers found in individual Y-shaped, can enter tissues and cause across the placenta. They activate the classical complement system and can act as to enhance phagocytosis. IgM is the first antibody produced during a primary immunity response, which is important, of course, for the diagnosis of a disease. It's a pentamer, so I would know the shape. It activates the classical pathway as well. IgA is a secretory ABS. Can exist as a monomer, but is mainly found as a dimer. I would know these shapes. So let me just go back to IgG. Can be found Y-shaped. IgA can be found as a dimer, sometimes a monomer, and IgM is found as a pentamer. IgD is only 0.2%. Its function is unknown. No one knows what the function is. And IgE, which is the least of them all, 0.002. This is mainly done with allergies and the allergic response. Like I said, it's 0.002. It's involved in vasodilation, defense against parasitic worms. So definitely know that. Definitely go to slide 14. It's a great chart. It's going to give you the percentages, the shapes, which know that 3 out of the 4 are monomers. So if you know what the pentamer is, which is IgM, and if you know what the dimer is, which is IgA, you're going to know that IgG, IgD, IgE are monomers. Know the percentage. Monomers are the highest with 80%, and IgEs are the lowest with 0.002%. No humoral immunity number 2. When it comes to the B cells, there's a pretty good chart or a diagram of the clonal selection and differentiation of B cells starting on slide 23. Once again, you have to know it. It goes in a nice little chart to help. It's nice and labeled. It's going to tell you every step you need to know. So that's your humoral, which are the B cells. Then you get to the cell mediated, which is your T cells. This is where it becomes a little hectic. This is where you're going to break into different T cells between your CD4 cells and your CD8 cells. Your CD4 cells are your helper cells, and your CD8 cells are your cytotoxic T cells, which are your killer cells. So CD4 cells are the commander-in-chief. The CD8 cells cannot work without your CD4 cells because the CD4 cells tell the CD8 cells, which are the killer cells, what to do. There are 2 classes of MHC. Class 1 are all cells except red blood cells, sperm, and ovum, and your cornea. Class 2 is activated by macrophages, mature B cells, and some T cells. So know your 2 different classes. There's 2 classes, class 1 and 2. And then it breaks down the helper T cells, which are the CD4 cells, and your cytotoxic cells, which are CD8 cells. Then there's the primary and secondary immunity response. Your primary is, of course, your first encounter with that specific antigen, and your secondary is the repeat encounter with the same antigen is responsible for memory. Because like I said, non-specific, which is your first and second line of defense, have no memory, but your third defense, which is specific, has that memory. So it's going to remember that antigen when it comes back around again. That is it, guys. I know that was a lot of information thrown at you. This is my longest video so far. I would like to wish you guys good luck. I know it seems hard. I know you don't think you can do it. Trust me. I know you guys have been studying. I know you guys are smart. It comes down to memorization, and I know that's hard for some of us, but some things we do, we don't know how or we don't like to, but we work a ways and we find a way to do it. I know you guys will. Once again, thank you for being patient. I wanted to have this video out sooner, but like I said, my Wi-Fi literally just came up when I was in Fairfax at 5 o'clock, and I had to drive an hour and a half to get here, have dinner, and I'm making this video. Remember that Dr. Kokash is having her review session tomorrow at 3 p.m. Check your email. She sent out, I believe, at the end of last week. If you cannot make that, my Piazza is open. I'm getting great responses from there. Like I said, I get the notifications, but since I'm the instructor, it comes in 4-hour increments. So if you post it, I might not get a notification until 4 hours later when it does its refresh and it sends me everything you guys sent. But I'm not getting to it. I don't know if it sends your classmates an email and your classmates just happen to go on Piazza because your classmates are answering your questions and I have not seen a wrong answer yet. I do go back through. I do say your classmate's right. Your classmate's right. Here's a little bit more information. I've been giving great links, charts to help you guys out. So of course, Piazza is there. Dr. Kokash is there. Remember, a Scantron, blue or green. Number 2 pencils. And remember to sit have a seat in between you and the next person. I will see you all on Thursday. Good luck, study hard, and just think, once this exam's over, there's only Friday, Saturday, Sunday, Monday, Tuesday, Wednesday, and then hopefully you're going to go have great turkey and great stuffing and mashed potatoes. And then just think, once the holiday's over, we're really close to Christmas. And of course that great holiday break where we can just put our minds on shutdown for a good month. So study hard, and I will see you guys Thursday. See ya.